Hi, everyone. This is Michael Lee. I'm the founder of the Data Incubator. We're a data science AI training and placement company. So if you are a company and you're looking to either hire or train data scientists, you can come to us. So we're probably best known for the fellowship that we run, where every quarter we have PhDs and master students who are looking to enter the industry, and they apply for this fellowship. It's completely free for the fellows who get in. And if you're interested, you can apply online at www.thedataincubator.com. We also have online courses for working professionals, and as well as corporate training options where we can go on site to your company or office. So if you're interested in those services, again, you can sign up at thedataincubator.com. But today we're doing another installment of Data Science in 30 Minutes. And this is a regular monthly series on data and its role in the world. And past speakers have included Kirk Bourne, who's the Director of Data Science at Booz Allen Hamilton, Holden Corral, an engineering lead at IBM and a core contributor to Apache Spark, Zubin Garamani, he's the Chief Scientist at Uber, Sam Swift, he's a VP of Data Science at Betterment and a former fellow from the Data Incubator, and many, many more. And if you'd like to see not just this episode, but some of the past episodes, you can go online to our website, thedataincubator.com, and you can view those episodes. Now, before I introduce today's speaker, I want to remind everyone that you can pose questions in the YouTube chat area. We'll be monitoring them, and the speaker will answer them at the end. You do need to be logged into your Google account to be able to pose these questions. And we will be monitoring them, so feel free to post them at any time throughout the talk. So without further ado, I'd like to introduce today's speaker. I'm joined today by my good friend, Ryan Copping. Ryan is the global head of analytics within the personalized healthcare organization at Roche Genentech. He's led this team of data scientists who support data science research and development by generating novel insights from real-world data sources, including electronic medical records, omics data sets, and clinical images. Ryan has really been working for Roche for the last 16 years, and he's held different data science leadership roles in the US and in Europe. Ryan's background is actually as a statistician and in computing, and he has a big passion for data and analytics, new technology, and understanding of fostering team culture and engagement. So without further ado, Ryan, do you want to take it from here?
Yeah, thank you, Michael. And thank you, everybody, for joining this call and hearing about what I'm going to talk to you about today, which is really talking about how data science is helping us advance healthcare, and in particular, our vision at Roche and Genentech around personalized healthcare. So what I'm going to do is I'm just going to jump over to my slides and give you a little bit of context, and then we'll dive into the content. At the end of the presentation as well, we've got some time for Q&A, so I look forward to the Q&A at the end. Just a bit of context before we dive into the content today. When we think about healthcare, data science is playing a key role across the healthcare industry. So for patients, providers, pharma, payers, and policymakers alike. What I'm going to focus in on today really is around the biopharma angle, and in particular, my experience at Roche and Genentech. Now, even within Roche and Genentech, there's data science being used across the board. So there's data science being used in diagnostics, in pharmaceuticals, in manufacturing, in all sorts of areas across the organization. What I'm going to focus on is my experience as well in research and development, and in particular on what we're calling personalized healthcare. So that's just a bit of context that this is a very broad topic, and I'm going to zone in on the area that I work in today. And I hope you find this useful, and I hope you find it interesting. Okay, so if we think about personalized healthcare, so at a high level, what we're looking to try and do with personalized healthcare is ultimately identify the right drug for the right patient at the right time. Now, in the past, our view of each patient has often been quite blurry, and that's what this blurry image represents. If we think about the depth of information and the data that we're collecting on the various patients, whether that's in a clinical trial or even in a real-world setting, it's meant that our view of each patient hasn't always been as clear as it could be. That being said, we've always still made good progress in terms of developing new medicines and new therapies to help support our patients. So even though we haven't had all of this data that gives us this level of granularity that we would like, we've often been able to develop really good medicines. But could you imagine if we had a much deeper and cleaner picture of every single patient? What might be possible then? That's where we think that we'd be able to get to this vision of getting the right drug for the right patient at the right time. So when I talk about a clearer view of each patient, what do I mean? So what we're really looking to do, and I'll share some examples with you in a moment of what we've actually done and some real tangible projects, is what we're looking to do is we're really looking to collect different data modalities on our patients so that we can understand each patient and their journey and their disease at an unprecedented resolution. So this type of information will include genomic information, clinical information, could be from the electronic medical record, or it could be from the clinical trials. Digital information could be from apps and sensors, for example, and then imaging. Images from clinical images, such as CT scans and MRI scans, but also digital pathology images as well. So if we did have access to all of that data, it would give us this higher resolution view of each patient and their disease. And what that ultimately will allow us to do is to start asking new questions of these data sources. And it will allow us to really reach this goal of identifying the right drug for the right patient at the right time. So we're calling this at the moment meaningful data at scale. And what our objective is here is to really be able to have access to data from a really diverse patient population that will cover enough patients to be generalizable, but also data that will be high quality as well. I think that's often a concern around some of these data sources. So another key part of this is also that we would like the data to be longitudinal. So we'd like longitudinal information on patients for all these different data modalities, where we also have clinical outcomes. Because if we have outcomes for the patients, what that will allow us to do is really understand the disease and patient heterogeneity when it comes to outcomes. So why are some patients and some subsets of patients performing better on certain therapies than others? Why might that be? So that's what we're really striving to try and address here. And what I'm going to show you in a moment is some initial projects that we've been doing this year and last year to really sort of try and make breakthroughs in the space. So obviously, data is the key here. I've just spoken a lot about data there. Historically, we've always captured data, and we've had data science being used in biostatistics and bioinformatics for many years to much success as well. However, what's changing now is this data landscape. So as we think about the past, which was really collecting data on a clinical trial where we maybe enroll a certain number of patients, for example, 200 or 300 patients, and we monitor them and we collect data on them on an ongoing basis throughout the trial. What we're moving towards and where we are today is to start to gather more and more information from outside of our trials, for example, from electronic medical records. And where we're trying to move towards is linking this data to other data modalities and other platforms that are maybe collecting data on patients as well. So as mentioned previously, that could include omics and it could include imaging. So there's a lot of challenges here, if we're honest, and we have a lot of potential and a lot of things still to overcome, but I think we're making some good progress in the industry here. And there's many companies out there that are doing very similar to Roche and Genentech, and there's many data providers out there as well that are making really good headway into this collection of data, but also curating and cleaning the data. That being said, there's still a lot of challenges. We really need to continue to work on data privacy and data access. We need to continue to invest in the curation of the data and the harmonization of the data. We need to continue to invest in the quality of the data and improving the completeness and the accuracy of many of these data sources. So we're by no means there yet, but I want to show you today that progress is being made, and I think this is quite an exciting field because each month that goes by, I feel like that we're making progress and enabling us to answer questions that we previously weren't able to answer. So just last bit of context before we dive into some examples. So if we think about developing new medicines, this is sort of historically what the timelines looked like. So, originally it was always a strive towards blockbuster drugs. However, as we started to really understand disease more and the heterogeneity of disease, and we're able to start looking into things like genomic testing, we were able to start developing more targeted medicines. So, for example, something like Herceptin for HER2-positive breast cancer. Where we want to move towards, though, now is really truly individualized treatments. Now, in order to enable this, we've spoken a lot about the data. That's pivotal. But once we have the data, what we really need there is we really need a talented group of data scientists to apply advanced analytics to this data to be able to uncover these new insights and these novel insights. And what's really important as well is to be able to interpret, is to be able to develop interpretable results as well, and things that we can then take back to the clinic, take back to our physicians, and take back to our drug developers to help us develop the right medicine for the right patient at the right time. Okay, so that's the context. So that's what personalized healthcare is, and that's what it means to us at Roche and Genentech. And as I mentioned, this is something that's certainly a hot topic within healthcare at the moment, and it's something that everybody's trying to do. So let's talk about, from a data science standpoint, what does the data look like? What do the applications look like? And also then from a technology standpoint, what are we using? It's probably going to be interesting for you to see what we're working with. From a skill set perspective, I know that's something that I often get a lot of questions around as well, is how may working in healthcare differ from other data science fields in other industries, for example. So I'll try and do my best to cover these topics. But let's talk a little bit about data first. So the first thing that we've been focusing on within Roche and Genentech, and again, a lot of other companies are as well, is trying to organize our internal data sets. So for tens of years now, we've been gathering and collecting data on patients in our clinical trials. In all honesty, these data have been analyzed for the purpose of that trial. Often we get the insights, we write up the reports, we write up the publications, we submit those to wherever they're going. It could be the health authorities, for example, to get an approval for a new medicine. But then we move on, and we move on to the next drug that we're developing. We move on to analyzing the next clinical trial. What we're starting to try and do now is have a much better concentrated effort of bringing together all of this data, the historical data we've got, harmonizing that, but also as new data is coming in from ongoing trials, making sure that not only retrospectively are we taking care of our data, but prospectively as well, automating as data comes in to make sure that it's being pulled together, harmonized, so that we can start analyzing data across the trials. And this is a huge undertaking. It's definitely challenging, but ultimately we want to strive towards that point of making this data available for research. And that's something that we now have done in 4 or 5 different disease areas now. But what I'm going to focus on is more our real-world data component, so the new types of data that we've been able to get access to, and I want to share some use cases with you on this. So the first example that I'm going to talk about is around what we call real-world data, so data from outside of the clinical trials. So there's an anecdote around the fact that generally, if you look at the US population and the patient population in oncology, for example, around 4% of patients are in clinical trials. So that means that for only around 4% of our US oncology patients are we actually getting access to their data and analyzing their data. Now, if you think about it, that means there's another whole, like 96% of patients out there where there's really important information that we could learn from. So companies such as Flatiron, which is a partner of ours, have been going out there and getting access to this electronic health record data, for example, curating the data and making it available for research to help the patients. So if we take Flatiron as an example, they have an electronic health record. Flatiron have a great team that curate this data. They go into the structured information from the EHR. They pull that together into analysis-ready data sets, but they also curate and clean up the data from unstructured fields as well. What they do then is bring this together into a research database that we can then access and analyze. And this is available to a number of different researchers across the globe, many biopharma companies, also academics, also FDA are working with this data, for example. So as of now, if we look at this data set, we have around 2.2 million active patients in the network from a number of different centers across the US. So I want to give you now just a couple of examples of the types of things that we've been doing with this data to give you a feel for it. Before I do that, I want to touch on one important topic that I get asked about a lot, which is, well, with data sources that aren't coming from our clinical trials, when they are coming from the real world, obviously they've been collected for different purposes. So there's a perception that the data quality might not be as high as a clinical trial. And in many cases, that is the case. However, I think what we're starting to see now is a lot of companies and a lot of institutions really play a heavy focus now on trying to improve the quality, the completeness, and the accuracy of their data to make sure that it can be used for research and to enable us to answer new questions. So just one simple example here that's been published, it was Flatiron's work on the date of death in their database. So as you could imagine, when we're analyzing data in the oncology setting, one of our key outcomes and key endpoints for our analyses is survival. So we're looking for the date of death. Now, traditionally, the date of death is not always captured very well in an electronic medical record. So what Flatiron have been doing, like many other different partners, is going in to try and figure out, are there better ways to start curating this data point, this date of death, populating this so that we can improve the accuracy. So without going into too much detail here, over the last couple of years, they've worked really hard to improve the sensitivity and the completeness of the date of death. And we've now got it to a point, for example, in lung cancer, where the completeness is now over 90%, which we now believe means that, okay, from a research standpoint, if we are to do a survival analysis, this can give us some robust results.
Yeah, that's really good.
So let's talk about let me give you an example of a survival analysis using that data now. So one thing that we'd like to be able to do with our clinical trials is really complement the data that's in our existing clinical trials with data from the real world. If you think about it, if we can get additional data from patients from the real world setting that already exists, that can give us additional information to make decisions on. So one of our ideas around this, and again, many different organizations and different groups are looking into this at the moment, one of our ideas is around building what we're calling external control arms. So the idea is, for example, we could have a single arm trial in oncology where we have our medicine that we're investigating. We could, for example, do a survival analysis. We could calculate the median overall survival, for example. What we could then do potentially is go into some of these real world data sources, build a cohort of patients that are similar to the patients from the trial. So we would use like an inclusion-exclusion criteria. We would use some statistical methods as well to match patients. And then we could do a survival analysis to try and understand how does the therapy that we've investigated in our clinical trial compare to some of the other standards of care being used in the real world in a similar group of patients. So we've been doing this exercise recently on replicating some of our old clinical trials just to try and show some validation that this could work. And here's an example of one of our pivotal lung cancer trials from our cancer immunotherapy drug, atezo, where we actually recreated the control arm and essentially got the same results when it came to median overall survival, and the hazard ratios were essentially similar. So this gives us this validation that we can start to go forward now, for example, with our single arm trials and potentially build these control arms out of different standards of care to help us make a more informed decision around how effective some of our medicines are. So that's one example. Another thing that we've been doing as well, which I think you might find interesting, is to start looking at, with this wealth of data, can we start to use all these different patient characteristics, so the clinical data, for example, along with the treatments they've received, the different diagnoses they've got, their medical history, their demographics, their labs, all of these different values that we can get out of the electronic health record. Can we start to build some algorithms that will help us predict outcomes for patients? So can we start to identify which patients, for example, are likely to survive longer than others? And then ultimately, why might that be? Are there some predictive or prognostic biomarkers that we can start to learn from this data so that we can design better trials and potentially target a more targeted patient population that's really going to receive benefit from the therapy? So one example that we just did recently, which I think is quite interesting, is we actually ran like an internal Kaggle competition, essentially. We've called it the Roche Advanced Analytics Data Challenge, the PAD Challenge. And we had 500 of our data scientists from across the globe within Roche, across 28 different countries, where we gave them a training data set of 7,000 patients from our Flatiron database. That was across 7 different cancer types. And we gave them the goal of building an algorithm to predict that the probability will be alive, that the probability a patient will be alive 1 year after their first treatment initiation. We then released a fresh data set of 3,500 unused patients. We asked everybody to make their predictions, and then we scored everybody's models. So this was an example where we were then trying to uncover certain biomarkers, but also, as well as that, it was a great exercise from a methodology standpoint as well, in terms of trying to understand what kind of advanced analytics methods worked in this setting. So as you might expect, just to give you a flavor, because I know one of the questions you might ask is around the methods that we used, for example. In this instance, the majority of the teams actually used decision trees. In this case, XGBoost was popular, as always, in Kaggle competitions, for example. And indeed, the winning team did use XGBoost. So gradient boosted decision trees were definitely effective. And we actually uncovered some really interesting results and things that we're now working with on our current projects that have given us a good feel for what type of characteristics may be prognostic when it comes to identifying if patients will have a good or a bad outcome on therapy. So I've spoken a little bit there about the EHR data. The next thing I wanted to talk about is linkage. I spoke at the beginning about the different data modalities and trying to link them together. Let me give you an example of that. So I think advancements are being made with data linkages. To be transparent on the challenges, again, there's challenges still around sort of the data quality, and that's constantly being worked on. And there's also challenges around the sample size as well and the size of this database. But I think as each month passes by, as all the infrastructure is in place, and as all these different organizations are working on improving the quality and capturing more data, I think the possibilities are becoming more and more fruitful, I think. And we're still at fairly the beginning of the journey, but if I could imagine fast forwarding to a few years down the line, I think we'll be in a really exciting place. So as of now, one example of this is we've actually, we've now been working, for example, with Flatiron and then Foundation Medicine. And Foundation Medicine have a next generation sequencing platform for sequencing patients and getting access to their genomic profile. We've actually got, there's a database that's now been brought together called the Clinical Genomic Database, which is the linkage between these two data sources. So you have the clinical data and the outcomes from Flatiron linked with the genomic data from Foundation Medicine. So again, I think this brings some interesting possibilities. So an example of analysis that we've done with this data, and I do want to set the context that this is still quite early days. This is something that we presented to ASCO last year. As the data grows, I think it brings more and more possibilities. But just to give you a small flavor of the kind of things that we could start to uncover if we have this rich information on patients. In lung cancer, we were trying to understand why there's a group of patients in lung cancer that we classify as rapid progressors. So these are patients that progress really quickly and the disease gets worse, regardless of which therapy class they actually receive. So you can see in the top curves there, you can see that there's the differentiation between the rapid and the non-rapid progressors. In 3 different therapy classes, you see the same pattern. So this happens. One thing we were able to do, though, was to start looking at, well, what about with the genomic data? Can we uncover potentially some of the mutational information and some of the genomic information that can help us give an indication of why this might be happening for certain therapies? And one thing that there's some promise on, and again, this has been looked at by many different groups at the moment, in and outside of the organization, is it looks like potentially there is a signature called tumor mutational burden, which is essentially a quantity of mutations. And it looks like when it comes to rapid progressors, the TMB value is lower in cancer immunotherapy in the rapid progressors than it is in the non-rapid progressors. However, in the other therapies, there doesn't seem to be that same pattern. So this has given us some indication that potentially tumor mutational burden could be a predictive biomarker for cancer immunotherapy. So I think that's quite exciting. And again, as the data grows, we'll be able to really start to dig into this. The sample sizes you'll see here are still quite small. This is from last year. And again, it goes with my messaging of we really need to continue to grow our data assets, improve the quality of the data and expand them so we can answer more and more questions. But again, I think this shows some exciting promise of this type of linked data source. Another example I'd like to give is actually with the images. So I mentioned linking clinical images to outcomes and clinical data as well. Here's an example of that. So this is an example where we've used a deep learning model on FDG PET images for a segmentation problem, essentially. Okay, so what we've been able to do here is we've been able to really go in and we've been able to build a deep learning algorithm which trained off a number of different images and trained against the ground truth, which is the radiology red image. so read by the radiologist. We've been able to train an algorithm that learns from that data and then is able to detect, from a deep learning algorithm, is able to detect and identify these different tumors, for example, and able to quantify the volume of tumor. Now, without going into a lot of detail, what we've done here is when we've gone in and done some validation on the test data sets that we've held out from different disease areas that we actually trained on, we've got accuracy results of around 90%, which we think is, again, super promising. This is quite early work, and it's actually going to get presented at a conference next week by one of my colleagues, Skander. Now, the interesting part of this is that when we actually compare radiologists, they actually are generally in agreement around 80% of the time. So it's definitely an improvement on that. So the value of something like this, where we could have an algorithm that could potentially identify and calculate the tumor burden, for example, this could help us not only in terms of efficiency and consistency of measuring disease burden, but it could also potentially be a prognostic signature as well. So it could be something that we could use then to try and understand for each different patient, depending on their result to that, what might their outcomes look like? Which group of patients may be at higher risk than others, for example? So I think this is, again, really sort of exciting work and just starting to demonstrate the possibility that may be possible here as we collect more and more images linked to the clinical data, linked to outcomes.
And what is the, how many images do you need to be in order to do this radiology example?
Yeah, that's it's $1 million question, I think, and depending on the different use cases we've been looking into, we do have access to different number of images. I think our answer is normally more than we've got. I think in this instance, you'll see our training data set here was around 2,200 different scans. And then our test holdout data set, we had two different disease areas. One was just over 1,000 patients, and 1,000 scans. Another one was just over 200. So I think in this use case, we felt we had sufficient data, and you can see there from the accuracy there of the test scores. But I think in some other instances we've worked on, we've come to the realization that we do need more images. So I think the key as well with the images is that we want to have access to images that are linked to clinical outcomes as well. And in some instances, it's quite easy to get access to images, but maybe not the outcomes. And I think that's another huge effort on our side as well, is getting these linked data sets. We've done efforts as well in ophthalmology, for example, where we've actually got a lot more images than we have in this example. So I think, yeah, it may be dependent on the question you're answering, but also the disease area as well and the type of scan you've got. Yeah.
Okay. Very interesting.
So there's some use cases for you. I know one thing that I was asked to talk about as well was around the toolkit. And one thing I wanted to share is I think historically in the pharma industry and in biopharma in general, and it's a bit of a generalization, we've traditionally tended to work with more traditional tools. So for example, SAS has been a big tool that we've used for many years, particularly on the clinical trial side. Now, there's often been good reason for that in terms of it being validated software, etc. However, as things have evolved in the last 5 or so years, I think the type of toolkits now that we're using are really expanding. And this is opening up more possibilities. It's also enabling us to work with much more diverse data sources. And I think from my standpoint, it's definitely very exciting to be involved in. So now I won't read out everything on this slide, but I wanted to give you a feel for the type of tools that we have in our toolkit these days. And primarily a lot of our work done in my team within personalized healthcare is primarily done with R or with Python. We have users of various other software as well. But also one of our big areas of focus has been around reproducibility as well. Obviously we're working in an environment where we need to ensure reproducibility. We are working with health authorities, etc. So that's where we've been really excited with some of the advancements with things such as containerization, for example. Even just in terms of the way the team interact as well now, I think, we've got a global group, half the teams in Europe, half the teams in the US. I think we're really trying to use technology to our advantage, like many other organizations and industries out there as well. So we do try and use things such as Slack and Discourse as well to help us enable that. So I think this has been really fascinating for me, and I kind of give an extreme example on the left, but certainly when I started in my career in biostatistics, for example, within clinical trials at Roche, that was really my toolkit. And then if I look across now to our toolkit now and what everybody's using, it's pretty exciting, and it's really enabling us to do interesting things, particularly, as I mentioned, reproducibility, but also in terms of the type of analytics we're able to do and the way we surface these analytics to our stakeholders as well. So really trying to make interactive analyses where we can, using things such as Shiny, for example, and R Markdown. So obviously technical capabilities are very important to be a data scientist, and I didn't want to dive too much into that. I've spoken about some of the toolkits there. Also, just from a quantitative science perspective and a methodology standpoint, obviously it's very important. However, I think there's also what's really important as well, I think, and where I see people be successful in our space is really around the mindset and behaviors as well, and some of the core competencies there. I think I wanted to just call out a couple of things that I think are really important from what I see, and many of these will be common to data scientists across all industries, but I think it's worth me giving my perspective from a healthcare perspective. I think curiosity is obviously a very important one. I think, as I showed, some of the data sources we're working with, they've often come from places where they weren't necessarily collected for research. So we really need to understand, how were those data sources collected? Why were they collected? Are there any potential biases in that data that we need to account for? We're working in healthcare. We're working to try and help the patients. We need to make sure that we're doing rigorous work, and that ties into this scientific rigor. I think it's really important from my standpoint as well, and from the group's perspective, that we really understand the data, understand the research question, and that we really take a rigorous approach to generate our insights. In addition to that, I mentioned earlier about having interpretability as well. I think that's also very important to us. If we really want to understand the science and want to be able to help the patients, we really need to understand and interpret the models, for example, that we develop, or the insights that we generate. Final slide now. Just final reflections around data science in healthcare from a personal perspective. I think for me, it's something that I've worked in now for around 16 years, and it's been really rewarding, and I really enjoy it. Because I feel like the goal that we're working towards of ultimately trying to improve patients' lives is a very important one. So I think in terms of the impact, I think as a data scientist, there is an opportunity to have a big impact. And as I shared with you at the beginning, there's challenges now around the data, and we'll come on to that in a moment. But I do think as we progress year on year now moving forward, there's a chance to really be unlocking the answers to some questions that we've never been able to answer before in the past. So the data, I spoke a lot about the data. I see it as the key. Progress is being made, but there are a lot of challenges. And actually, I see this as exciting challenges. Sometimes it can be frustrating, but actually it needs data scientists like ourselves to be able to go in and to understand how we can improve these things and what we would need to improve in order to enable the type of research we want to tackle. So again, I see that as an opportunity. But we also then have to be realistic. So from an advanced analytics standpoint, there are things we would love to be able to do today, and questions we'd love to be able to answer where we just maybe don't have the data, and maybe we don't have the data, or maybe we haven't even curated the data yet to be able to answer the question, because the data curation and cleaning the data, that's a huge effort. However, there are examples, like I've shown you earlier, where we are making progress with that, so where we are able to tackle the questions. But I think one thing that's important from my standpoint in healthcare is around applying the right approach to the right question. It's not always about trying to go in there and do some machine learning. I think, it's about taking the right approach. We've done many studies, and we've been doing a lot of methodology exercises where we've been comparing some of the latest machine learning approaches, for example, with some of the more traditional approaches, for example, logistic regressions, etc. And in many cases, they will still outperform some of the newer methods. That's not to say they always will. It may be an indication of where we are in terms of the data sets today, but I think it's really important we take the right approach and the right method for the right question. I also spoke about interpretability earlier, but that's usually important, particularly as we start to try and feed information back into our research and development efforts. Interpretability is key. There may be some healthcare use cases where interpretability is less important, and we may be interested in a pure prediction question, for example, for clinical decision support, but I think that's probably a little more rare. And in terms of a career, just from my standpoint, I think, getting to work on interesting research questions and things that really matter to the world and to the patient population, I think that's really rewarding, but also being able to continuously develop your skills, whether that's learning new data sets, learning new methods, learning new technology, but also working really closely with our stakeholders and with, for example, the clinicians to really understand the science and try and understand what questions are that they'd love to be able to answer that we could maybe try and help them answer. From my standpoint, I think that's something that's really exciting as well. So obviously biased, but I think working as a data scientist in healthcare is something that's super exciting. Okay.
Wonderful. Thank you so much for that, Ryan. So you talked a lot about growing, sort of needing a larger data set. Can you say a little bit more about that? What's holding you guys back from getting more data sets? Is it access to the actual data? Is it sort of the need to just sort of clean and work through the data you already have access to? Where's the bottleneck for you guys?
Yeah, there's a couple of different angles to it. I think, so sometimes it's around getting access to the data. So sometimes data is collected, but for whatever reason, maybe privacy perspectives or whatever, and there may be situations where we're not able to use that data for research. However, there are now more and more situations where we are able to get access to the data from research perspectives. The challenge then becomes the curation of that data. So cleaning the data up, curating it, harmonizing it. Often you're collecting data from a lot of different sources where you need to try and harmonize it so you can analyze it together. As well as that, I think the different data modalities I mentioned about really having this complete view of a patient where you'll have the clinical data, their outcomes, their omics data, their images, their digital data. I think we're really quite at the infancy there of being able to link and connect all those data sources together. In many cases, maybe one or two of those modalities is being collected. It's rare that all of them are. So there's a lot of efforts now to start collecting all these modalities together and do more sort of prospective data capture it there to sort of have these data sets that are both deep and rich on each patient. So I would say in terms of the data modalities, I think that's starting to come together in terms of the ability to link data sets together. But when you think about the scale of data sets as well that are needed sometimes, so we're trying to do a lot of things in, for example, rare cancer types. You may have 2 million patients in your network, but once you go in and try and identify the patients with lung cancer who've also got a BRAF mutation and have also been treated by treatment X, your sample size is dwindling down and down and down. So I think the scale of the data as well, just as we go through the next 2, 3, 4, 5 years, I think will continue to grow and grow and will enable us to start looking at these more niche populations.
Got it. So a question from the audience, what are the main data science techniques that a person who is interested in healthcare analytics should learn?
Techniques. Okay, so I mean, I think from my standpoint, a lot of the examples I was sharing there, we do a lot of, for example, survival analyses. So that's definitely something that I think is important, regardless of the disease area as well. For the sort of bread and butter type of analyses we do today. I think moving forward now, I mentioned there some of the things we did, for example, where we're trying to predict patient outcomes. I think that's where we're looking at maybe more some of the more newer techniques. In many cases, as I explained, some of the more traditional sort of statistical learning and statistical modeling techniques often perform pretty well in the size of data sets we have, but we want to keep pushing the boundaries there as well. So we do want to continue to invest in that space as well.
And you were talking about the importance of interpretability. Are there techniques that you wouldn't use because the interpretability of those techniques is not quite there?
Yeah, definitely. I think so. We're often sort of a bit nervous. Not only, so I'll talk about the interpretability of the results as well, but I think we also often want to really try and understand how something's working as well. So if it is a black box model, sometimes that may make us a little bit nervous, just again, because of what we're trying to do with healthcare. We're trying to support patients. We're trying to really understand disease heterogeneity. I think it's really important we try and understand what's happening with the data. So I think, but from an interpretability standpoint, I think it's really important to us. We want interpretable models, and we've just done an exercise in my group where we've been doing some benchmarking of different models, both sort of black box and white box, if you like, models, tried to understand the pros and cons of each. And I think interpretability always comes back as such an important factor, particularly when we want to sit down with our clinicians and talk things through and share some insights and generate some hypotheses for the next analyses.
Got it. And are there any tools, techniques, or methodologies that you like to use when you're having to link together different types of data sets? So image data with clinical data with genomics data. Are there things that you find that are particularly good at doing that?
Yeah, so I think so. If you talk to our, I mean, if you look at our guys who are doing a lot of our clinical imaging work, you know, they're heavy Python users, in many cases using TensorFlow as well. I think as we start to link multi-modalities together, I think a lot of the challenge is actually around the curation of the data. For images, there's a lot of curation effort as well, starting from anonymizing the images all the way through to actually analyzing them, but also around sort of linking these data sets together and cleaning the different data modalities, bringing them together. Yeah, so I think it's kind of quite diverse. And then I think for a lot of the work we do with the clinical data, that's where our more traditional sort of packages will be used. You know, a lot of our users, as I mentioned, are R, for example, using R, for example, our real world data users, they'll be using tidyverse, for example, and using the various different packages that come along with that. They'll be using, for survival analyses, you know, survival is the main R package that we would use for that. And then as we start to look at more of these prediction type questions, that's where I've mentioned things like XGBoost, for example. Yeah.
Got it. And as you're thinking about sort of using image data, I know one of the concerns, especially around data leakage, is you could have, for example, two types of CAT scans coming from two different locations. And one location, for whatever reason, happens to have a higher instance of the disease you're trying to detect. It turns out that the one place that does happens to have a green stamp on the bottom right-hand corner for their logo for their company, and the other one has a red stamp on the bottom right-hand corner for the logo of their company. Obviously, a human looking through this can sort of know to disregard that data, but when you're looking through this, the TensorFlow is going to pick that up and be like, oh, you know what I think is a really great factor? Whether or not there's a green logo on the bottom of their of this image, right? Like sort of stupid artifacts like this that a human would put into a model, but would be easy to pick up. Do you get a lot of that? How do you think about dealing with it?
Yeah, it's a really good point. I think so. The example I shared with you today, and a lot of the examples we're working through are with that from our own clinical trials, where we're collecting a lot of images on patients on a daily basis. I think there we have certain standards when it comes to the capturing of the images and the curation of the images to try and harmonize and to make sure we don't fall into those challenges. I think in all honesty, where we're more concerned, and I guess not concerned is maybe the wrong word, excited as well, is getting access to the real world images. And we've been getting access to those now. And I think that's where things really get interesting, because you're going to have images, could be, MRIs, for example, could be consistent from that standpoint, but using from different machines, from different centers with a different resolution.
Exactly.
That's where things get really challenging. And I think that's why a lot of our effort is around the curation of the data. And that's the big bottleneck and the big challenge. Is around the creation and harmonization of that data. And that goes for the clinical data as well. I spoke a lot about that, but certainly for the imaging data. And obviously as well, even just from a privacy standpoint as well, making sure that we've curated that data so that we're de-identifying it as well. That's super important. So we actually work with external partners, for example, to do that work in many cases.
Oh, wow. Oh, okay. So they sort of de-identify, but obviously you want to keep the linkages between the medical records and the images, but interesting.
The challenge is we don't just want access to images. As I mentioned in my slide, it's about this linked information. So we need to have a patient identifier that we can link, and then we can see the outcomes, we can see the clinical data as well.
Got it.
And then
okay.
Of that, Michael, as well, is kind of, that's sort of, that's work that's evolving every week at the moment. So we've got our imaging folks in data science, in personalized healthcare, where I am, and they're making great progress every week, but every week as well coming across new challenges and new issues. So I think I can imagine another year or two down the line, we're going to have some amazing success stories. I think the imaging, has got a lot of promise when it comes to data science.
Wonderful. Well, it was a real pleasure speaking with you, Ryan. And for those of you who are interested in learning more about either what Ryan is doing or what companies in general are doing, you can go to our website, thedataincubator.com, and you can find more of our video series, Data Science in 30 Minutes. And if you're interested in learning more about either our free data science fellowship for PhDs and master's students, or our online courses for working professionals, or our corporate training offerings, you can also go online to thedataincubator.com. Thanks so much, Ryan.
Thank you. Thanks.